this video, let's talk about bacterial cell structure, in particular the cell envelope and external structures. The cell envelope refers to the plasma membrane, the cell wall, and in some bacteria, an additional membrane. While I admit this might not sound riveting at first, it's actually really foundational knowledge to understanding how pathogens work, how they infect us, and how we as humans fight back. So if you're like me and you love understanding how stuff works, for example, why you would take one antibiotic over another, then this information is for you. Because we've all experienced illness from bacteria. I'm sure I'm not the only one who's had questionable leftovers. Or perhaps you were an unlucky soul who ate at a restaurant chain when they unknowingly had contamination due to some local outbreak. We can even expand this further to things like UTIs and sinus infections. In many cases, the culprit is bacteria. To understand how tiny microbes like bacteria can make you feel that bad, we need to look at their armor, the cell envelope, and external structures. Actually, these structures are often targets for drugs like antibiotics, meaning the very structures that might allow a bacterium to invade your body could also make them susceptible to our drugs that fight back. So starting with the outermost layer, this bacteria might wear a glycocalyx, or an external layer of polysaccharide that extends from the cell surface. There are two main types of glycocalyces, capsules and slime layers. Let's start with the capsule. Capsules are well-organized layers of the cell envelope external to the cell wall and cell membrane. They are most often comprised of polysaccharides, and for some bacteria, they are physically anchored to the outer cell membrane, and therefore are not easily washed away. An example of a capsule-containing bacteria would be Streptococcus pneumoniae, and I think you can guess the illness that this particular bacteria causes. Without a capsule, these bacteria will not likely get you sick. However, with a capsule, Streptococcus pneumoniae can evade the immune system and resist phagocytosis, which is when immune cells basically Pac-Man chomp microbes that shouldn't be there. So bacteria can evade phagocytosis when they use their capsule like camouflage. Capsules also contain a great deal of water, so in other environments, this layer can help prevent a bacterium from desiccation, which is another way of saying drying out. They can also be protective against most hydrophobic, toxic materials like detergents, and can even keep the bacteria safe from viruses, because yes, bacteria also get infected by viruses. The other type of glycocalyx, the slime layer, is more diffuse and unorganized compared to the capsule. It's water-soluble and can be easily removed, not the case for the hardy capsule. You'll see that there's a common theme when it comes to these external structures. They're usually going to help the bacterium either 1, stick to something, or 2, move towards or away from something. The slime layer is cool because there's actually instances of both. Gliding bacteria produce slime, which has been shown to facilitate motility, and motility is just a fancy word for movement. That slime layer can also, not surprisingly, help bacteria stick to surfaces like tissue surfaces in plant and animal hosts. Next up, we have pili, which in the singular is pilus. These structures are used by many bacteria kind of like Velcro, so they might help bacteria attach to the lining of your stomach, or in the case of some E. coli that cause UTIs, the bladder. Without these pili structures, these bacteria would probably be washed out of your system before they could cause trouble. The terms pili and fimbriae are often used interchangeably. However, there are slight differences, and specifically, the term fimbriae usually is used to refer to these projections that aid in the attachment to surfaces, like helping a bacteria stick to a rock in moving water, or stick to your gut lining like we mentioned. Pili can be evolved in attachment, but also motility and even the uptake of DNA. This has to do with horizontal gene transfer, which is something really interesting, and we'll get to it later. And then there's flagella, tail-like motors that let bacteria swim. For example, Heliobacter pylori uses its flagella to move through the stomach into the mucosal layers of the stomach lining, where it can establish itself and cause inflammation and even ulcers. Fun side note, for the longest time, people with stomach ulcers were told that stress was the main culprit for their pain, so they were often given stress reduction tactics and medications that didn't really do much. It wasn't until two physicians, Dr. Warren and Dr. Marshall, had the idea that perhaps these stomach ulcers were often caused by bacteria, and in particular H. pylori. Now, the really wild part of the story is that Dr. Marshall actually used himself as a test subject to see if this was true. He inoculated himself with H. pylori, developed a stomach ulcer and gastritis, which is basically inflammation of the stomach lining, and then used antibiotics to treat himself and healed. At first, there was some opposition in the medical community whether this was valid, but eventually in 2005, these two won a Nobel Prize. So even before a bacterium gets inside your body, these structures are already helping the bacteria do a lot, helping them stick to stuff, move around, and evade your immune system. But those aren't the only structural layers that matter. Let's take a look at what's happening just below the surface. When trying to identify an unknown microbe, one of the first lab tests to run is a gram stain. This quick staining procedure divides bacteria into two major categories based on one key feature, their cell wall. Almost all bacteria have a cell wall made of a tough, net-like material called peptidoglycan. Gram-positive bacteria have a thick layer of peptidoglycan. That's what holds onto the purple stain during the test. Gram-negative bacteria, on the other hand, have a thin peptidoglycan layer and an extra outer membrane on top. It's like a cell wall sandwich where the membranes are the bread on each side. After the staining series, these gram-negative cells end up pink. So how does it work? The gram stain series starts with a purple dye called crystal violet. We stain the cells on the slide, wait a minute, and then wash off the excess stain. Next, we add a mordant, iodine. This helps lock in the dye for the gram-positive cells, making sure they hold on to that purple color. Actually, the iodine forms large crystal violet iodine complexes inside of the cell. Next up is the decolorizing step. Right before this, everything is purple from the crystal violet stain. When we add the decolorizer, though, it causes all of the gram-negative cells to lose the purple color. Why? Well, let's take a look at the structural differences to understand. So for gram-positives, they have one cell membrane and one thick layer of peptidoglycan, the cell wall. The alcohol, or decolorizer, dehydrates the cell wall and causes its pores to close, preventing that dye-iodine complex from being washed away. Gram-negatives, though, they have this thinner peptidoglycan layer and an outer membrane containing lipopolysaccharides. Gram-positives don't have this second layer. So, the lipopolysaccharides in the outer membrane dissolve readily and allow crystal violet to wash off in gram-negative bacteria. So, that crystal violet stain washes out, leaving the cells colorless. However, bacteria with no stain at all are really tricky to see under the microscope, so we've got to do something for those gram-negatives. Therefore, the last step is a counterstain. To counterstain, we'll add safranin, which will color all the gram-negative cells pink. The gram-positives are also hit with safranin, but they already are saturated with the crystal violet, so they just remain purple. While the terms gram-negative and gram-positive are widely used to describe these cell structures, I actually prefer the use of the terms monoderm for gram-positive, referring to their single cell membrane, and diderm for gram-negatives, since they have two cell membranes in their cellular envelope. I want to touch on two important characteristics that help us distinguish between our gram-positive monoderms and our gram-negative diderms. Our gram-positives contain techoic acids. Remember that they only have one cell membrane and a cell wall, so the techoic acids help anchor the cell wall to the cell membrane. Our gram-negative diderms contain lipopolysaccharide, often shortened to LPS. Now, something really important is that LPS is an endotoxin, meaning that it triggers the human immune response. If many gram-negative cells die off in the body all at once, say from antibiotics, then a lot of LPS can be released all at once. Lipid A, a particular portion of the LPS molecule, is toxic to humans. As we mentioned, LPS is considered an endotoxin. So we can see a really severe immune reaction, and in some cases, it can even put a patient into shock. So already, we can see why this information matters. Differences in cell types between bacteria can affect everything from how bacteria cause disease to how we treat it, and even the symptoms. So now that we've got the basics, let's take a look at how this microscopic armor can actually be used against pathogens. Or, in other words, how antibiotics can punch holes in those defenses, sometimes quite literally. If we know whether an infection is caused by gram-positive monoderms or gram-negative diderm bacteria, we can choose the right tool to fight back if we need to. In cases where intervention is necessary, It means choosing an antibiotic. The true definition of an antibiotic is a natural product or the derivative of a natural product that kills susceptible microorganisms or inhibits their growth. However, definitely note that in today's language, when we say antibiotic, we're typically referring to a drug that specifically targets bacteria, and the term alone can sometimes refer to both natural and synthetic compounds. A more inclusive term you might hear is antimicrobial. Antimicrobial drugs include both naturally and chemically synthesized compounds, and the term is not specific to any one microbial group. So an example of an antibiotic is penicillin. It's the first true antibiotic to be discovered and produced on a large scale in the 1940s. It's actually derived from Penicillium fungi and is effective against gram-positive bacteria. Because it's derived from a living organism and is effective against bacteria, it's considered a true antibiotic. There are many antibiotics that work by targeting the cell envelope, especially the peptidoglycan wall. Penicillin actually blocks bacteria from building their cell wall. This is like poking holes in a balloon that's already under pressure. So what do I mean by this balloon analogy? Well, bacteria typically face constant osmotic pressure due to the concentration of solutes inside of the cell typically being higher than the concentration of solutes outside of the cell. If you remember the basics of osmosis, this would cause water movement into the cell. The cell wall helps bacteria resist that osmotic pressure. So without the cell wall, the cells will literally explode. Think about that if you ever need to take a penicillin class antibiotic. One reason you don't exactly feel amazing when taking them is because your immune system is dealing with the cleanup of a bunch of exploding cells. I invite you to pause and try to figure it out. Which cell type is penicillin most effective against? I know that no one ever actually pauses, but seriously, try it. Penicillin works especially well on gram-positive bacteria, since their cell wall is thick and exposed. Gram-negative bacteria have kind of a secret weapon, their outer membrane. Remember that phospholipid membranes found in bacterial cells and human cells alike are selectively permeable. Not just anything can waltz on through and get past this barrier. So this extra layer blocks many antibiotics from even getting to the wall underneath in those gram-negative cells, especially if the antibiotic has a relatively large molecular structure. So to treat gram-negative bacterial infections, we might need a different type of drug, like a polymyxin, which can go after that outer membrane directly. There are, of course, other drugs that target those previously mentioned structures, like the capsule. We now know, for example, that if the capsule is compromised, the bacteria's ability to evade the immune system and establish infection would probably also be hindered. That's why it's so important to know about bacterial structure and organization. If you use the wrong antibiotic, it might not even reach its target. And beyond obvious reasons for wanting to take an effective drug and not wanting to take an ineffective drug, proper diagnosis and prescription is actually so important for not just human health, but environmental health on a larger scale. What do I mean by that? Well, the large-scale, unnecessary use of antibiotics not only contaminates the environment, it actually leads to resistance, meaning that the drugs that we use could become ineffective in the future. When we overuse antibiotics, say, as is really common in the meat industry, those drugs end up putting a selective pressure on the microbial populations that live there. So any microbe that happens to have a leg up on survival will survive and reproduce, and it changes the gene pool. Some ways that bacteria exhibit resistance is by producing enzymes that actually dismantle the drug, or modifying their cell walls so that the drug is no longer effective, and even pumping the drug right outside of the cell. All right, so antibiotic resistance is a really complex problem. So for now, just take away that although we cannot stop resistance from occurring, we can definitely slow down the process by being really intentional about how we use antibiotics, both in the medical community and agricultural sectors. We can slow down resistance so that we can buy time to actually use these drugs and save lives. It's better for humans and also lowers contamination in our environment. So whether it's food poisoning, skin infection, or something more serious, understanding the bacterial cell envelope and external cell structures helps us have a good grasp on what we're dealing with, whether we need treatment, and the right tools to effectively get better.